Hello student, good morning. To continue the lecture, that is mutation. In mutation, germ, tract and mutation. It has been noticed in Drosophila and most other animals that only those mutations that occurs in the germ tract can be transmitted to the offsprings. In this way, one gets the impression that the mutations occurs only in germ plasm or germ tract, but in mosaic mutations have been seen to occurs in the body cell, that is somatic cell or body soma. It therefore can be concluded that mutations are not linked to the germ cells or germ tract, but these cells are more susceptible to mutation in comparisons to other parts of body. Once mutation occurs, it is multiplied by the cell division and passed in numerous sperm or egg cells. There are different examples of mutation as dominant gene. There are different examples of mutation as dominant gene caused chondrodystrophic dwarfism, in which legs and arms are extremely short and deformed. Its mutation rate is 0.0070 and it appears in every 4,300 gametes. The recessive gene caused mutation is the hemophilia, where blood clots very slowly. In mutation rate is 0.0032 and it appears once in every 31,250 gametes. Properties of mutations. Mutation never arise. Mutations never arise gradually, but it appears suddenly. It appears in a single individuals and transmitted to next progeny. Mutation may be morphological, physiological or biochemical. Mutation in Drosophila in such characters like fertility, longevity, serological properties and behaviors. All mutants, whether from prominent or inconspicuous mutation, always belong to the same biological species to which the ancestral form belong. Example: wings, eye or eye color in Drosophila. Induced mutation. The mutation causing mutagens were discovered by Muller in 1927, where he successfully induced mutations in Drosophila by exposing to X-rays. After them, may experiment on plants and animals have successfully been conducted with the help of X-ray and other high energy penetrating or ionizing radiations. There are some other agents which may induce mutation are eosin, anthracene, inflorescent light, mustard gas, ethyl urethane, which may induce mutation or erosion. Anthracene, inflorescent light, mustard gas, ethyl urethane, manganese salt, ultraviolet rays, and every high temperature in any case. However, X-ray, Y-rays, and cosmic rays are the chief tools with investigators to induce mutation. In plant and animals, certain chemicals are said to produce mutation in certain way and are non-mutagenic when administered in other ways. Example, formaldehyde with food is mutagenic to Drosophila. Gene mutation. The gene mutation is the result of small, sudden, spontaneous and random change in the genes. The new gene, that is mutant gene, results in a altered character. Most of the variations arise in this way. Since these mutations include very limited segment of DNA, these are called point mutation. The point mutation may be of the following types: substitution mutation, frame shift mutation, gene mutation, substitution mutation, frame shift mutation, transition and transversion, replacement of pyrimidine by another pyrimidine in a polynucleotide chain caused by tautomerization, ionization, base analogous diminution, transversion, replacement of purine by a pyrimidine or vice versa is caused by alkylating agents, depurination, that is the substitution mutation. Frame shift mutation, deletion mutation and insertion mutation. Two types. Number one, substitution mutation. In the substitution mutation, a nitrogenous base of a triploid codon of DNA is replaced by another nitrogen base or some derivative of the nitrogen base, changing the codon. The altered codon may code for a different amino acids and may result in the formation of a protein molecule with a single amino acids substation whose effect may be seen in altered phenotype. The substitution mutation may be of following two types: transition are changes that involves replacement of one purine in a polynucleotide. Transition and ionization. First is transition. Transition are the change that involves replacement of one purine in a polynucleotide chain by another purine and correspondingly in the complementary chain in the replacement of the pyrimidine by another pyrimidine. The transitional substitutional can be introduced by any of the following ways, either during DNA replication, copy error mutation, or otherwise. Tautomerization. Tautomerization. In a normal molecule of DNA, the purine adenine A is linked to the pyrimidine thymine T by the two bonds, while the purine guanine G is linked to pyrimidine cytosine C. However, all these four nitrogenous bases rarely exist in the alternate state. These states are called tautomers and are formed by the rearrangement in the distribution of hydrogen atoms. Tautomeric shift due to the tautomerization and amino NH2 group of cytosine and adenine is covered into the amino NH group, and likewise keto group C double bond O or thymine and guanine is covered to enol group OH. In its rare tautomeric state, a nitrogenous base cannot pair to its normal partner. Rather, tautomeric purine adenine pairs with normal cytosine and tautomeric guanine with thymine. Similarly, tautomeric thymine pairs with the normal guanine and cytosine with the adenine. Such pairs of nitrogenous base, known as forbidden base pairs or unusual base pair. These rare base can introduce mutation during DNA replication. Tautomerization. Second is ionization. Transmission may also introduce by ionization of the base at the time of DNA replication. Ionization involves the loss of hydrogen from number 1 nitrogen of nitrogenous base. For example, in its ionizing state, thymine pairs with the normal guanine and ionizing guanine links with the normal thymine. Third, base analogues. Certain chemical compounds have molecular structure simple to the nitrogenous base present in DNA nucleotides. These are called as the base analogues. Deaminization. Certain chemicals like nitrogen and NH2 hydroxylamine, nitrosomethyl, urea, NMV change the base sequence in DNA by the series of chemical steps. These cause the deaminization of nitrogen base replacing amino group NH2 by hydroxyl group OH. The deamination of cytosine leads to the formation of uracil. Deamination of adenine forms hypoxanthine exhibits similarly with guanine. Second is transversion. These were first populated by E. Fries in 1959, in which type of gene mutation alkaline agent like ethyl methane sulfate EMS and methyl methane sulfate MMS, including substitution by the two ways, by substitution by purine for purine or pyrimidine for pyrimidine transition. Second is by substitution of purine for pyrimidine or pyrimidine for a purine transversion, that is the changes A double bond T or C double bond G. This is the types of mutation. Okay, thank you.